Hello students, welcome to Educational Avenue. So today we will start a chapter in biology that is fundamental unit of life. That is basically about the cells. Like what is cells, what are the component of cells, how the cell is made up of, and some scientists that were involved in the discovery of the cell. So we will start the chapter by the scientist names who has discovered the cells. First is the Robert Hooke. He discovered a dead cell. First scientist. First scientist was Robert Hooke. He discovered the cell in 1665, and the main is he discovered a dead cell. Just keep in mind that Robert Hooke discovered a dead cell. Second scientist came, that the name of that scientist is Anton van Leeuwenhoek. He discovered the cell in 1676. And he discovered a living cell after the discovery of microscope. This time, at the time of Robert Hooke, microscope was not there. Then the theory came. That theory is the cell theory. In that cell theory, three scientists were there. Three scientists were involved. Skleden, Sackvon and Virchow. First two scientists, they gave three statements that all organisms are made up of cell. Cell is the basic structure and all cells arises spontaneously. These three scientists, they gave three statements. All organisms are made of cell. Cell is the basic structure. And third is spontaneously. Then this third scientist, the name is Virchow, he expanded this cell theory by stating that all the cells arises from the pre-existing cells. Virchow gave a statement, all cells arises from pre-existing cells. Means a cell is there, by the help of that cell, the another cell will form. This is the cell theory that is given by these three scientists. First, second was the Skleden and Sackvon, and the Virchow expanded this theory after them. Now we will see about the plasma membrane. We already know that plasma membrane is the outer boundary of the cell. Means we all have a house. All the houses are having their outer boundaries so that it will have a protection. Like this is a cell. Center is the nucleus, this is the nucleolus, and different components will be there. So basically First we will see about the plasma membrane. This is the plasma membrane, this structure. Outer boundary is the plasma membrane. If we will zoom this structure, like if we are expanding this structure here, it will have a bilayer structure of lipid like this. And this will continue like this, and it will form a complete boundary. I'm just expanding this structure of this plasma membrane here. So this plasma membrane is having the outer boundary. Why the plasma membrane is there? You just see the structure. It is separating the outer environment. This is the outer environment, okay, and this is the inner environment. This plasma membrane is separating with these two parts. So this is responsible for the partition, or you can say the chemical reactions, whatever that is happening inside the cell is not mixing with the outer environment. Okay, there are many functions. One, I have said this, that chemical reactions will take separately from the outer environment so that the reaction can take place. Then this plasma membrane will always means this plasma membrane is having some pores, means you can say this plasma membrane allow selective molecules. Because of that selective nature of the molecule, anything cannot go inside from the outside and from inside to the outside. Suppose this cell wants A molecule from the outside, and the outer environment is also having the B molecule and the C molecule. So cell wants this A molecule. So this is the plasma membrane. This plasma membrane will allow only the A molecule inside of the cell, and because of this the A will go from outside to inside. But at that time the B and C will also try to get inside, but it is not possible because at that time the cell only needs A. Because of this reason, the plasma membrane is also known as selective permeable membrane. Means it is having a selective nature. Anything cannot cross from the plasma membrane. The molecule which the cell only wants, that will cross inside or the outside. So this was the function of the plasma membrane. There are many functions of this plasma membrane, like you can say the osmosis process will happen by the help of this plasma membrane only, and there are cell receptors on the plasma membrane that will allow, means the same what I have said, the movement of the molecule. The receptor, if the receptor will be there, they will allow. Cell signaling is the function of plasma membrane. These all are the function of plasma membrane. Now we will see organelles. Cell organelles. What are cell organelles? These are the components present inside the cell. Component present inside the cell, or more specifically you can say in cytoplasma. Cytoplasma is the liquid part. This was the cell. Inside that center nucleus is there, and this is the cytoplasma where all the reactions was happening. So in this cytoplasma only these cell organelles will be there. So cell organelles, we will see there are many cell organelles present in this cells. So we will see line by line. Like we cannot write the cell organelles in any order. First we will see the nucleus. Okay, you can see the structure here. I will draw a different diagram. In that you can see clearly, like I will draw the structure inside the cell, and by the help of that I will explain the cell organelle. This was the cell. Center is the nucleus, and inside that nucleus, nucleolus is there. Okay, first we will see about the nucleus. Okay, nucleus is called the brain of the cell. Human body having the brain. What is the function of the brain inside the body? It controls the functions, it controls the coordinations, whatever we are doing in day-to-day life. Just like the same, nucleus is the brain of the cell. It is a present center. Okay, if we will talk about the animal cell, in animal cell the nucleus will be on the center, and if we will talk about the plant cell, it will be on the peripheral side. So just we will take we will take the example of animal cell. Nucleus is called the brain of the cell. This nucleus is having a double membrane layer. In the diagram you can see it is having a double membrane walled structure. So this is the nuclear membrane. These nuclear membrane will have some pores for the exchange of materials. Like suppose here the DNA is there. Okay, from the DNA protein is getting synthesized. So this protein we need to transport it outside. So this nuclear membrane will have that nuclear pore. This is the nuclear pore. Okay, this nuclear pore will allow this protein to go outside. So this is the structure of the nucleus. Outer is the nuclear membrane that will have the nuclear pore for the exchange of materials. Inside that the nucleolus will be there. Okay, now we will see the next organelle. That is the endoplasmic reticulum, that we can write as ER. Endoplasmic reticulum. Okay, so it will continue from this only. It will be continue from the nucleus only. The structure you can see. Okay, you can see here. Now there will be two types of endoplasmic reticulum. One will be the SER and another will be the RER. This SER, you can see here, it will be like this, smooth in appearance. Smooth in appearance. In RER you will see a rough structure on the surface. This is the structure of RER. Okay, so this will have a rough appearance. So this is the basic difference between the SER and the RER. This S stands for smooth and R stands for rough. Smooth in appearance, rough in appearance. So there are different functions of these two. This RER will always be related in the protein synthesis. Protein synthesis. And this SER will be involved in lipid and steroid synthesis. They both are having their different functions. Okay. First was the nucleus, then it will continue to the endoplasmic reticulum, that will be having two parts, SER, this RER. Okay. Now we will see next organelle. That is our Golgi body. This organelle is discovered by Camelio Golgi. Okay. Now here we will draw the structure. Complete structure I will draw here. It will be like this. So this is the diagram of Golgi body. Okay. This is known as the cisternae. This is called tubule. So this will have means this Golgi body is responsible for having some functions, means it is inside this cell. You can see, I will draw the diagram here also. So the function of this Golgi body is modification, packaging and storing. Modification, packaging and storing. This is the main function of the Golgi body. Suppose here the protein synthesis takes place, or in this RER, we already know that RER is involved in the protein synthesis. From there, the protein will come to the Golgi body, and that protein will come inside this Golgi body, and that protein will get modified, and after modification it will go under the packaging, and by the help of these vesicles, these are known as the vesicles, these small are the vesicles. Inside this the protein will be there, modified protein will be there, so it is helping in that function. So this vesicle will have the protein. Okay, modifications means mainly you can say like a defective protein is there or incomplete protein is there. So by the help of this Golgi body, the Golgi body will modify or convert that inactive protein to the active protein into the vesicle, and that vesicle will provide to the cell wherever it will need inside the cell. So this was the function of Golgi body. Now we will see our next cell organelle, that is lysosome. So this lysosome is also present in the cytoplasm, means this is the area of the cytoplasm. In this cytoplasm only the lysosome will be there. Like here you can say like this, like this, like this, like this. So this lysosome, suppose I am drawing a structure here. Inside the lysosome it will have hydrolytic enzyme. Hydrolytic enzyme it will have inside the lysosome. So what is the function of this hydrolytic enzyme? This hydrolytic enzymes will basically digest all the thing whatever it will come inside from outside. Suppose here food is there, the food will go inside in this lysosome, so it will get digested by the help of this hydrolytic enzyme. This is the function of the lysosome. This will have a membrane, outer boundary will have the membrane, and inside that the hydrolytic enzyme will be there. This hydrolytic enzyme will have low pH, means concentration of H plus will be more. Okay, because of the presence of H plus ions, the enzyme are hydrolytic. Okay, and it will have low pH. Now one of the major function or event you can say we can see in this lysosome, like what happens now when the lysosome when a cell will get defected by anything or by any chance, that cell, suppose this cell get defected, okay, this is a defective cell. So inside this cell the lysosome is there present already. So here the lysosome will do one function, that this lysosome will digest this whole cell, because there is no use of this defective cell inside the body. So there is a reason it will digest this whole cell. So that is called suicide bag. So the lysosome is also called the suicide bag. Why? Because if a cell is getting defected, then the lysosome will digest all that cell. So that is called the suicide bag. The name suicide, you can understand that someone is doing the suicide means die. So this is the one of the major function of the lysosome. So next we will see our next cell organelle, that is mitochondria. This mitochondria, here is the diagram. Okay, this is the outer membrane, and this is the inner membrane. By this you can say mitochondria is a double walled organelle. This mitochondria is an organelle that is having a double layer of wall. So it is double walled organelle. Inside this the matrix will be there, and these curves are known as cristae. Okay, now we will see the function of mitochondria. So basically this mitochondria is responsible for aerobic respiration, you can say. Here in the inner mitochondrial membrane, means this inner membrane will do the function of aerobic respiration. Okay, so inner membrane will do the function of aerobic respiration. Because of that, ATP production will takes place. Okay, so what was the function? I'm rubbing this. So it is having a function of aerobic respiration, then ATP production. Okay, this mitochondria will have their own ribosome. Ribosome, DNA. Okay, so this is mitochondria. Here I will draw the structure. This is the mitochondria. Now we will see another cell organelle, that is plastid. Plastid, this is mainly of three types, you can say: chloroplast, chromoplast, and leucoplast. Okay, so in this main is chloroplast that we have to see. I will draw the structure of this chloroplast. Okay, this is the basic structure of the chloroplast. This is the thylakoid. This complete, you can say, grana. This is the stroma lamella. Okay, this is the structure of basic structure of a chloroplast. Now, what is the function of this chloroplast? This chloroplast is present in all the green plants, that helps in photosynthesis. That helps in photosynthesis. Because of that photosynthesis, the plant produces their food. So that is one of the main function, you can say, of the plastid. The plastid also help in the storage of foods. If you will take leucoplast, that is the third part of this plastid, it will have three parts: amyloplast, aleoplast, and proteoplast. Proteoplast. These are the three types of leucoplast. This helps in the storage. Okay, amylo for carbohydrate, aleoplast for oils, you can say. Proteo, the name itself, it is saying the protein. These are the three forms of the leucoplast. So this is all about plastids. Just a rough diagram I will draw here. Now our last is vacuoles. This vacuoles means what? Empty space. So suppose this is a vacuole. So this space should be filled with something. So this is for storage purpose. Okay. So the main function of this vacuole is for storage purpose. Like waste, you can say, whatever the waste of the cell is there inside the cell will get stored in this vacuoles. Now one of the major function of this vacuole, you can say, like this vacuole will be filled with water. Now this cell is having a vacuole. Now it will apply a turgor pressure all around the cell. Turgor pressure. Because of this turgor pressure, it is keeping the cell in shape. So this is the main function of the vacuoles. Just I will draw. So these are all about cell organelles. So I hope you understood very well about the cell, the fundamental unit of the life. Thank you.